Welcome everyone. I'm so grateful that you are listening to my lecture. I'm Raoufi from Dr. Shafiq High School and Isfahan Province, Iran. Cancer is the second leading infectious disease for fatalities in the world. And many people die because of cancer. Many environmental issues have an effect on cancer. And cancer can cause many environmental problems and pollutions because of the fatalities. So now, my method is a novel method for early diagnosis of cancer in early stages. It's a novel method for biosensor fabrication based on target recycling and polymerization signal amplification strategies. First of all, we want to know what a biomarker is. Biomarker is a natural occurring molecule, gene, or characteristic by which a particular pathological or physiological process, disease, or etc. can be identified. By measuring it in the blood, you can identify and find out about a biomarker that can lead to diagnosis of a disease. For example, the biomarker, the glucose is the biomarker for, as you know, diabetes. Then we want to know there are some usual ways to diagnose the disease. Usually cancer, PET scan, CT scan, MRI, PCR. But they have two disadvantages. First of all, they have a long time of diagnosis and they have low sensitivity. Then we want to know what an electrochemical biosensor is. The most used biosensor is a self-contained integrated device which is capable of providing specific quantitative or semi-quantitative analytical information using a biological recognition element. Biochemical receptor, which is retained in a direct spatial contact with an electrochemical transduction element. Then there are some ways of increasing signals, actually novel way in our project to increase the signal in electrochemical biosensors. It's DSN, duplex specific nuclease, and ATRP, atom transfer radical polymerization. Then we want to know each of them and how do they work. Duplex specific nuclease is an enzyme purified from the hepatopancreas of red king Kamchatka crab. It's a kind of crab, as you know. And moreover, the enzyme is capable of discriminate between perfectly and non-perfectly matched short DNA duplexes. It actually cleaves with DNA with DNA or DNA with RNA double strands, as you know. And now, what is ATRP? Atom transfer radical polymerization is an example of reversible deactivation radical polymerization. Like its counterpart ATRA or atom transfer radical addition, ATRP is a means of forming a carbon-carbon bond with a transition metal catalyst. And then we want to know how our biosensor works. Biosensor, our electrochemical biosensor is made of 3 electrodes. The working electrode, reference electrode, and counter electrode. The most important electrode is actually the working electrode, that our surface is made of AU, gold, as you know. And now we have 3 amounts of targets, that is microRNA 21, that is the biomarker of breast cancer in the place. Here we have a non-amount of target, and we have medium amount and high amount. And here, as you see, first of all, the surface of our electrode is empty. Nothing is on it. Then we put hairpin DNAs on the surface. As you see, they come to the place. So we don't have any target, as we said. When we have no amounts of target, you see nothing. It's microRNA 21. RNA is completely matched with these DNAs. They don't create a bond. Then we put a ZR, that is our metal catalyst, in the place. Then BPAA, that is our better say initiator, comes to the place, and then with EATRP, as we said, we put our monomers, label monomers, that create a signal in the place, and our polymerization starts, and with SWV line graph, as you see, we have high amounts of signal. Here we have medium amounts of targets. As you see, like before, our surface is completely clear, working electrode. Then we put hairpin DNAs on the place, and then we have a medium amount of target, not much but a medium concentration. It's microRNA. You see it creates a bond. So what happens next? DSN, as we said, it's this enzyme cleaves DNA with RNA strands, double strands, and as you see, two of these electrodes, hairpin DNAs, go out of the place and far from the electrode. Then our metal catalyst come to the place. BPAA, as we said, is our initiator, and then EATRP comes to the place, and we have less hairpin DNA, so we have less amount of signal, and then SWV line graph, we have medium amount of signal. Then here we have high amount of targets. Hairpin DNA's surface is clear, hairpin DNAs come to the place, and then we have high amount of targets, so actually all of these hairpin DNAs become bonds and matches with these microRNAs, with these hairpin DNAs, and then DSN cleaves them. They go far from the electrode, so as you see here, there are no hairpin DNAs left. When ZR in metal catalyst come, nothing is on the surface. Then AU, we have here BPAA initiator, nothing in the place, and then SWV line graph, we have no amount of signal because we have nothing, no hairpin DNAs on the surface. So as you see, those dots in our line graph creates a calibration diagram. That here we had no amount of signal, medium amount of signal, and here we had high amount of signal. You see a decrease in this calibration diagram, and by measuring the signal according, we can find actually that how much is the concentration of biomarker 21, microRNA 21, that is our biomarker in the blood. So we want to prove that our biosensor works right. Here we have, as you see, we have some line graphs in this first part. As you see here, it shows that the obstacles that are in a place that let our actually electroactive to come to the surface. When we have no obstacle, you see we have an almost straight line graph like A. As our performance of our electrochemical biosensor goes on, you see a less amount. You see we have more obstacles. It curves. We have more obstacles, it curves more, and when DSN comes, it cleaves them. They go out. It's straight again. Here we want to show that, and the thing we are talking about is right. It's some microscopic pictures. Here we had just the hairpin DNAs on the surface. And you see, we have not much, the place is not crowded. Here we had lots of RNA, microRNA, and DNA create bond here. And here, when DSN comes, you see it's less crowded again. Here, we want to show that when some important factors in our biosensor isn't there, you see the cause it has on the signal. Here, when we have actually said, when we have no amounts of targets, you see in E, this orange line graph, we have high amount of signal. And as it goes on, for example, when we don't have DSN, we have no signal, but when we don't have hairpin DNAs, we have no signal, or when we have a medium amount, you see in this purple one, we have medium amount of signal. Then it is exactly what I said, different amounts of targets in the place. When the target increases, the signal lowers, as you see. And this signal creates a calibration diagram here that by measuring the amount of, actually the signal, logarithmic calibration diagram, by measuring the signal and by measuring the amount of signal we have, we can find the concentration. First of all, and in this last part, we want to know that our biosensor actually works. Here we have non-complementary microRNAs. Here we have double mismatch and single mismatch. As you see, in this non-complementary double and single mismatch, we have a little drop in our amount of signal, but not much. But when we use completely matched, that means microRNA 21, the target is in the place that comes with our hairpin DNAs, and they create bonds. You see a sudden drop in the signal, and here it shows exactly that our biosensor works. Here we in normal human serum and buffer phosphate serum. Here we have 1 nanomolar 1 picomolar and 10 femtomolar. Here, as you see in the human serum and buffer serum, we have a little bit difference in amounts of signal, but not much. Why? It exactly shows that our biosensor works. In human, we have actually a little more signal because we have lots of things in the blood. But so the difference is not much, but so it's not important, it's not significant. That shows our biosensor works. And in this last part, we want to know that we had some difficulties in using and creating this novel method for biosensor. First of all, in this first part of it, we have a very little amount of and concentration of actually microRNAs for all kinds of cancer in the blood. So we need some ways to create a large amount of signal. How we did that? That's simple, by using ATRP. It's a novel method. We don't use ATRP for biosensor, but we did it. And for another thing, that the biomarker and the biomarkers, microRNAs, are very small in size. So we need something, and we need to track them down in our biosensor and in the blood. So how we did that? By using hairpin DNAs and creating match. That's it. Thank you so much for listening to my lecture about biosensors.